Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the complement system. And in this particular video, I want to focus on the third type of the complement system, which is known as the lectin complement pathway. This was the classical and the alternative pathway that we discussed in our previous videos. We discussed in detail about the classical pathway and we discussed in detail about the alternative pathway. Uh, to start this particular video, I want to give you an overview, or I would say a recap of the nomenclature of the complement proteins. That these complement proteins are designated by the capital letter C, which is followed by a number from 1 to 9. For example, the C1, the C2, C3 till the C9, and these numbers are actually showing the order of their discovery. These complement proteins, they circulate in the serum in inactive form and they come into effect only once activated. What that means that they are there in their inactive form, but when you need them, they get converted into their active form. Now, for the activation of these inactive complement proteins, the complement proteins, they are cleaved, and the resulting peptide fragments, they are denoted by adding a lower suffix of A and B. When you look at these fragments after the cleavage of the complement proteins, so in most of the cases, the smaller fragment resulting from the cleavage of the complement protein is designated by a small a, and the larger fragment is designated by a small b. C2 protein or C2 complement protein is an exception in which the situation is the other way around. Now, the complement system, in my previous video, I've told you that this is part of the humoral immunity. This is part of the innate immunity. And we call this as the complement system because it complements the ability of the antibodies and the phagocytic cells, thereby helping the immune system. And the complement system is responsible for clearing the microbes and damaged cells from our organism. When you talk about the complement system, it consists of a number of small, inactive, and liver synthesized protein precursors circulating in the blood. And there are about 50 proteins and protein fragments, including the plasma protein and the cell membrane receptors. I've told you that there are three biochemical pathways that can activate the complement system. We discussed in detail about the classical complement pathway. We discussed in detail about the alternative complement pathway. And this particular video is going to focus on the lectin pathway. When you talk about this lectin pathway or the lectin complement pathway, if you are going to start the lectin complement pathway, so as you required in the classical pathway that there would be an antigen antibody complex. In case of the lectin complement pathway, you do not need any antigen antibody complex to start the lectin complement pathway. When you talk about this particular pathway, first you need to understand what are lectins. These lectins, they are actually the carbohydrate binding proteins. And when you talk about this particular pathway, the lectin complement pathway, so the lectin that is used in this particular pathway, that is known as the mannose-binding lectin. For short, we call this as the MBL. So this would be a specific lectin responsible for the activation of the lectin complement pathway. Now, this MBL, as I've told you, that this is synthesized by the hepatocytes, of course, the liver cells, and the MBL is released in the bloodstream in the inactive form. And of course, when you need them, you are going to activate this. Now, the the MB lectin pathway, that is responsible for the activation of the MB lectin complement pathway, and this is the structure of the mannose that is required in this particular pathway. So whenever the mannose binding lectin, it finds a mannose in the structure, it is going to bind over there and it is going to activate the lectin complement pathway. So how the system works, let us discuss about the mannose or the presence of this particular carbohydrate in the different pathogens. For example, if you talk about the Salmonella species, they are responsible for causing diarrhea, the typhoid fever, the septicemia in the human beings. And if you look at a specialized species from this particular genus, the Salmonella enterica, it have got particular protein which are known as the fimbrial adhesins, and these familial adhesins are responsible for adherence to the host cell. If you look at the structure of this familial adhesins, they have got mannose in their glycan structure, and thereby the MBL can recognize this and it can go for the activation of the lectin complement pathway. Let me give you another example. If you talk about the genus Neisseria, they have got many important species like the Neisseria gonorrhoeae that is responsible for causing the gonorrhea, and you have got the Neisseria meningitidis, which is responsible for causing the meningococcal disease. If you look at the this particular Neisseria species, they have got a specific surface protein which is known as the Neisserial surface protein A. This particular protein is involved in the bacteria's ability to adhere and invade the host tissue, and it have got a glycan structure that include the mannose residues. So if I've got the mannose, you can use the MBL to activate the lectin complement pathway. Let us take an example from the fungus world. You have got the Candida albicans responsible for causing the cutaneous candidiasis. And if you look at the cell wall of the Candida albicans, they contain mannose-rich glycoproteins, and they are responsible for immune evasion and the adhesion. And the MBL that can actually activate, that can actually recognize these mannose, and then they can activate the lectin complement pathway. So these are the targets where the MBL can bind and can activate the lectin complement pathway. So how the pathway actually starts? Say, for example, if this is a fungus, let us say this is Candida albicans. So on the surface of the Candida albicans, you have got glycoproteins, and they contain the mannose residue over there. When there is mannose on the surface of the Candida albicans, the MBL protein that is going to interact with this particular mannose, and when the MBL that interacts with the mannose, it is going to recruit two other proteins. One is known as the MASP1, the other one is known as the MASP2, and this MASP is actually the MBL associated serine proteases. So this M would be for the MBL, the mannose-binding lectin. This A is for the associated, this S is for the serine, and this P is for the protease. So when the MBL it interacts with the mannose on the surface of the pathogen, it is going to recruit the MASP1 and the MASP2. Now, in this particular complex, you have got the MASP1 in the inactive form, but when the MASP comes in interaction in this particular complex form, the MASP1 is going to go for the autoproteolytic cleavage, and because of the autoproteolytic cleavage, the MASP1 that is going to move into the active form. When the MASP1 it gets activated, the substrate for the active MASP1 is the inactive MASP2 in this particular complex. And when the MASP1, the active form of the MASP1, it acts on the inactive MASP2, it is going to cleave and activate the MASP2. Now you have got the MASP1 and the MASP2 in their active form. Now, when they get activated, they are going to make a complex. So you are going to get a complex of the active MASP1 and 2. And this active complex of the MASP1 and 2 is responsible for the cleavage of other complement proteins. This particular complex is going to cleave the complement protein 4, the C4. And when it acts on the C4, it is going to give you C4A, and it is going to give you C4b. So the C4 have been cleaved into these two fragments, the C4a and the C4b. Now, this active complex of the MASP1 and 2, it also acts on another complement protein, which is known as the C2. And when the active MASP1 and 2, it acts on the C2, it is going to give you two fragments. One is known as the C2a, and the other one that is the C2b. As we are talking about the serine proteases, so you can expect that in these particular proteins, the MASP1 and the MASP2, their active site should contain a serine because they are serine proteases, and serine is a very important component of the active site of the serine proteases. So the active complex of the MASP1 and 2 have given you four fragments, the C4A and the C4B from the C4, and the C2A and the C2B from the C2. Now, what happens is that the C4B from this particular the cleavage of the C4 and the C2A from this particular cleavage, they are going to get combined with each other, and this is going to give you the C3 convertase of the lectin complement pathway. Now, the C3 convertase, which contains the C4B and the C2A, that is going to act on the C3 protein, the C3 complement protein. And when it acts on the C3 complement protein, it is going to give you the C3A fragment, and it is going to give you the C3B fragment. Now, the C3b fragment that is going to combine with the C4b, C2a, and the C3b, and this particular complex of the C4b, the C2a, and the C3b, that is actually the C5 convertase of the lectin complement pathway. Now, as the name indicates, this is a C5 convertase. So this particular C5 convertase is going to act on the C5 protein. When it acts on the C5 protein, it is going to cleave this into two fragments, the C5a and the C5b. From this particular complex, from these particular fragments, the C5b is going to interact with another complement protein, which is known as the C6 complement protein. So now you have got a complex of the C5b and the C6 protein. Now, all of the complementation reaction till the binding of the C6 to the C5b, that is going to take place on the hydrophilic surface of the target membrane. And when the C5b that interacts with the C6, it is actually going to start the formation of the membrane attack complex that you are going to see in a while. If you look at over here, I've got the C5b, the C6, but this particular thing, they are still on the surface of the membrane. When you add the C7 to this particular complex of the C5b and the C6, you can actually see the transition. They are moving from the surface into the cell membrane. So they are moving from the surface into the interior of the cell membrane. Now, to this particular complex of the C5b, C6, and the C7, now you are going to add another complement protein, which is known as the C8, and it is still, you can say, allowing the transition of this particular complex from the surface into the cell membrane. At the end of the day, the C5b, the C6, the C7, the C8, they are going to combine with another complement protein, which is known as the C9, and this whole complex is going to make a pore in the membrane of the pathogen. And this particular pore, it has got a size of 70 to 100 angstrom in diameter, and this is going to move from one end to the other end of the target cell. Now, when the spore is formed, what is the disadvantage to the pathogens? Now, this particular pore formation is going to allow the ions and the small molecules to diffuse freely across the membrane, because the permeability of the membrane that has been compromised due to the formation of this particular pore. Secondly, this is going to lead to the disruption of the membrane integrity. So the membrane integrity of the pathogen is compromised, leading to the leakage of the ions and the small molecules across the membrane. Now, when there is pore formation, that is going to lead to the loss of essential electrolytes from the microbial cell. This is one damage that is caused. Secondly, this pore is also allowing the inflow of the extracellular fluids like the water, ions, and other small molecules into the microbial cell, which is going to lead that the microbial cell cannot maintain its osmotic stability, and eventually the cell that is going to get lysed and that is going to get killed. So this is how the lectin complement pathway it works.